Okay, think we're ready to go. Can people online here?
Yeah, I can hear.
We have a little bit of fan noise in the room, so I'll try to speak up.
Yeah, he's studying it down.
Okay. So we're going to continue with relaxation theory. Why does that show in? That's odd. Okay, now maybe we're good to go. Well, just to refresh your memory from last week, we derived an equation for the density operator in the interaction frame, sigma 2 for T, by analogy to what we did with the random phase model. And the critical assumptions were the same, and the most critical one being, the two most critical ones being that we're interested in times much longer than the correlation time of the stochastic process, which is safe for rotational diffusion, but maybe not safe for other dynamic problems. And then that we could patch the results that would predict that the density operator relaxes to zero by adding in the Boltzmann equilibrium density operator as an ad hoc correction to ensure that the density operator relaxes back to Boltzmann equilibrium. And as I said, we can check this assumption by doing the full quantum mechanics or by doing NMR experiments. So we have to transform back out of the interaction frame to the lab frame. And we want to address the fact that we know that the Hamiltonian could have terms with different rotational properties in it. Because we've already seen in the CSA Hamiltonian that there's parts of the Hamiltonian that depend on I minus and IZ and I plus, and these all rotate differently. And that's different from our simple random phase model, because in the random phase model, we were just talking about fluctuations along the z-axis, so rotations around z were irrelevant, and now they're relevant. And as I said, what I'm going to do here initially is different than what's normally done in textbooks, including my own. So I have to fix it in the next edition. So we want to transform back out of the laboratory frame, and we start that process easily enough. We have an expression for the differential equation in the laboratory frame. We and differentiate, we can solve this equation for the density operator in the lab frame rather than the interaction frame. And that's shown here. And the first term, of course, we just recognize that this is just the commutator of H naught acting on the density operator. And that's simply all of the terms that would come from the chemical shift, Hamiltonian, the scalar couplings, all the parts that survive isotropic averaging. So that part is simple. And then the second part here is the inverse transformation of the differential equation for the density operator in the interaction frame. And now I just substituted in our equation. That interaction frame equation was the integral of the correlation function of the Liouvillean for the fluctuations in the interaction frame. And then I unpack the exponential of the Liouvillean superoperator, because that's really one of these sandwich operators with one term on one side and the inverse on the other. So the second line shows that, where now I have e to the minus IH naught operating on the left and e to the plus IH naught operating on the right. And then on the third line, I first notice that I can move this operator inside the integrals and the additions. And now I've inserted a bunch of unity operations, identity operations, again, in the form by putting in the operator and then its inverse. I've done that here. I've done that here, and here's the part I started with. And this is because my transformations are based on these kinds of sandwiches. So I want to figure out which ones I can do right from the start. For example, this first one, e to the minus h naught t, L1 is in the interaction frame at time t, e to the IH naught at time t. All the time variables are the same. So I know that's just the inverse transform from the interaction frame back to the lab frame. So I immediately can write this first operation as just L1 in the lab frame times t. And similarly on this side, I just have the same time variables, t, something that's independent of time, and the other operator t. So this I can transform back, and I just have the density operator in the lab frame. So those parts are easy. And then I'm left with the transformation in the middle, where the time variables are different. So I have to do a little bit more work. But we're only going to worry about stationary processes. So we only care about the time lag tau, the difference between t prime minus t. I'm going to substitute into this equation for t. I'm going to substitute in that t is, let's see, t prime minus t is tau, so t is t prime minus tau. So I'm going to substitute in for t here and here. T prime plus tau. There might be a minus sign wrong here. I'll fix it. I substituted in, and now in the unitary operators here, I have a T prime and a T prime. So I can do that transformation. And that will bring L1 in the interaction frame at T prime back into the lab frame. And at the same time, I'm remembering that T prime is T minus tau. So I've substituted that in. So I'm back into the lab frame, and now I have the transformation with respect to tau of the superoperator at T minus tau. And to make any progress, now we have to actually look at the Hamiltonian. We've gone as far as we can, just manipulating the symbols. To go further, we have to know the Hamiltonian. So apart from looks like I've defined here, I'll have to check, look, I defined tau as T minus T prime rather than T prime minus tau. It'll turn out not to matter because the autocorrelation functions are even in time. And I'll try to fix it and make it consistent.
That's the limits integration in computation. You need to change that. Like tau is 0 makes it T.
So if E prime is T minus if tau is T minus T prime, when I change the integrands, this should have become positive. Is that what you're asking? I probably have a sign error in here. I'll fix it. And now we go back to what Ann talked about in slightly different notation, is decomposing the Hamiltonian in terms of the product of a spatial tensor and a spin tensor. And as I said here, we mostly care about k equals 0, 1, or 2. The order of the tensors. For relaxation theory, I also pointed out we have another problem, which is we have to keep track of the difference in operators that have the same coherence order, Q, but have different eigenfrequencies. For example, S plus and I plus both have coherence order of +1, but if they're nitrogen and hydrogen, their frequencies are very different. So their rotational properties around the z-axis are very different. So we introduce another variable P, whose only purpose is to keep separate operators with the same coherence order Q, but different eigenfrequencies. And the eigenfrequencies are the eigenfrequencies of the commutation superoperator. And in fact, the way the spin operator got defined was to make the spin operators an eigen function, an eigenoperator of the commutation superoperator. That's what defines them. And we wanted to define them this way because that means that the interaction frame transformation is very simple. It just introduces a complex exponential phase factor. Because if we expand the exponential in Taylor series, we just end up then term by term with an exponential in omegas, or Taylor series in omegas that we recognize as the exponential of I omega t.
I'm a little confused why you choose the base operators being eigen operators of the operator I just said. So
Right. It's because I wanted this expression at the end. Again, it's always helpful to know the answer. So I have to do this transformation in and out of the interaction frame. I want that transformation to be as simple as I can possibly make them, make my life easy. And the simplest it could be is that I just pick up a phase factor. And to pick up just a phase factor, I have to define the spin operators to satisfy this equation. So I don't know how Redfield thought this up the first time around, but knowing the answer, I can work backwards to what I have to do. If I chose something else, for example, then what I would see is the interaction frame transformation was going to generate some linear combination of all the basis operators. And that would be messy. Right?
if it's an eigenoperator of the Hamiltonian, though, it would still be an eigen, like it would still be an eigenoperator of the Lewis. Right?
That was my question also, if the Lewis is constructed out of the Hamiltonian. Anything parallel to the Hamiltonian, be parallel to me? If not, why not? You can add other terms once you make it into a Lewis and then you spoil. You know, Lewis can be something other than a rotation, dissipation and whatnot. That's it.
I don't know, I have to think about
I guess I'm just like, because if you just use the basis operator of the Hamiltonian, then you wouldn't have to introduce this extra something over P as well, right? Because
Oh, no, that would still
that would
come.
still be.
That would still be, you would still have to keep track of different frequencies.
Okay.
Yeah, but the expansion, the spin operators might look a little different, but you would still have the same problem because Q, right? So for example, if K is 2, Q takes on 5 values, but there's more than 5 eigenfrequencies in the spin system. So there's going to be operators that have the same Q but different eigenfrequencies. Other questions? That could well be, I mean, H naught here in principle contains the Zeeman and chemical shift and scalar coupling Hamiltonian. The reality is, at least in solution NMR at high field, it's just completely dominated by the Zeeman and chemical shift Hamiltonian. So the scalar coupling isn't very important. So for humbling problems, dipole interactions, CSA, quadrupole interactions in solution, the interaction frame transformation is just a rotating frame transformation. And in that case, it may well be that the eigenoperators of the Hamiltonian or of H naught are the same as the eigenoperators of the commutator. But I don't know if that would be true if we added in other terms than the Hamiltonian. I have to think about that. There was, and we'll remember a student named Oleg Trott many years ago, and every time I would write an equation down and say this was true, he would say, that's true in 3 Cartesian dimensions, but not in 5 dimensions with skewed coordinate systems. So could well be that there are generalizations or exceptions. So here, just to remind you, was an example. Of the chemical shift anisotropy, Hamiltonian, and then if we do the transformation into the interaction frame, nothing happens to Z, that's a rotation of Z around Z. I plus picks up a phase factor minus I omega IT. I minus picks up a phase factor in the other rotation sense. So in the interaction frame Hamiltonian, we have something that looks very similar, except we have these phase factors that depend on this particular frequency. We don't have the problem of P in this case. We just have Q, omega of Q. So this is what we wanted to transform. And now we're going to plug in this expression for the Hamiltonian in the transform frame for H1 here, and then do the transformation term by term. So here's the transformation of each operator by H naught of T. So we pick up the phase factor, and we're back in the lab frame with this phase factor. So now we can write everything down. We have then double sums over Q and Q prime because we have to, back here, the Hamiltonians inside here are already back in the lab frame, but we have to express them with the tensor expansion as well as this. So we end up with substituting everything in a double sum over Q and Q prime, a double sum over P and P prime. We have the double commutator now of the operators because the spatial tensors are just numbers. So they can come out of the commutator just like they're just multiplication factors. So we have this part of the expression of the double commutator, and we have this. Now, a general, and remember in these F's are basically a bunch of physical factors like bond length and chemical shift anisotropies and the spherical harmonics. And it's a general property of the spherical harmonics that these will be uncorrelated unless Q prime is equal to minus Q. So that one of these sums collapses. And this property is basically because if you think about, for example, Y2 naught only depends on theta, and Y21, say, has a dependence on theta and phi. So these extra Z rotation terms end up imposing this requirement that you have to have the Z rotation in one spherical harmonic has to be exactly canceled by the Z rotation in the other. Otherwise, the integral around phi is going to kill the terms. So that brings us to this. By the way, what's this integral? What kind of mathematical operation is it? Well, this is an autocorrelation function in the brackets.
Yeah.
And then, but what's the integral? If you're, it's a Fourier transform. Remember our simple analysis of the random phase model to get the adiabatic part of R2, we integrated the autocorrelation function. And now in the general treatment, we see we need to Fourier transform the autocorrelation function. But if omega is 0, this reduces to the integral. So our adiabatic treatment, our simple treatment, captured the part of relaxation where omega is 0. That's the adiabatic part. There's no precession, no. And now our full treatment captures everything. So we have the autocorrelation function. It's Fourier transform, but we only need to know the value of the Fourier transform at specific points, at the eigenfrequencies of the operators. So the Fourier transform, right, in general, is a continuous function. We just need its value at some discrete points. That's, of course, kind of a weakness in trying to study dynamics, because we want to go backwards. We want to measure the rates and look backwards through the Fourier transform to the autocorrelation function, and then learn about the dynamics from the autocorrelation function. But to look backwards, we would need to know the values of the Fourier transform at all frequencies, right? It's not enough to know. You can't undo an integral only knowing a few points in the integral. So that's one reason, by the way, that we're interested in relaxometry, where we can get many, many more magnetic fields, because that fills in more and more of the Fourier transform, essentially. And this part, this double commutator, right? So the result of the integral is going to be some number. And since these are going to be related to relaxation, if this integral is a big number, you expect that relaxation rates are going to be large. And if this Fourier transform gives small numbers at the designated frequency, the relaxation is going to be slow. That's a numerical factor. Here we have the double commutator, and that might be 0. So if that's 0, that contribution to relaxation is 0. It doesn't matter how big the integral is. The integral could be huge, but there still won't be relaxation. It's forbidden essentially by the symmetry of the spin operators. So I always think of this as being like a selection rule in infrared spectroscopy. You have infrared spectroscopy in some class. You learn that there are selection rules that indicate whether, based on symmetry properties, a particular transition can be excited by infrared irradiation. And if not, there will be no absorption, even if you match the energy. You can match the energy of the photons to the energy gap, but you still won't get absorption because the absorption process is symmetry forbidden. And that's kind of true here. If this double commutator goes to 0, then we won't get relaxation. It's forbidden because of the properties of the spin operators. So what we call the power spectral density is the real part, and that's going to drive relaxation. The real part then is going to end up if we were to solve the differential equation, giving terms that look like e to the minus something t, and that's going to be a damping. That's going to be some kind of relaxation. This result depends on Q, but luckily in isotropic solution, all the real parts of the spectral density are related to each other just by some other factors of -1. So we really just have to write or calculate one spectral density function rather than 5. So that's just a convenience. On the other hand, you have to make sure you keep track of all these -1s to the huge power that keep popping up, because if you get them wrong, then you're going to be adding terms when they should be subtracted or vice versa. So the first dozen times or so, I actually tried to do this derivation, then calculate a number. I got crazy numbers just because of the -1 getting mixed up. Now, the imaginary part is called the dynamic frequency shift. And Raphael Bruchweiler showed many years ago that you could recast this dynamic frequency shift term into something that looked like a commutator, and therefore it could be lumped in with H0. So dynamic frequency shifts are shifts in the resonance position caused by dynamics. But they're very hard to measure. And mostly we just say, well, the resonance frequencies are a little bit off from what we really think they are, just based on chemical shift, and we don't worry about it. Every so often, somebody writes a paper trying to measure them, but it's just hard to measure them accurately enough to make use of them. Anish?
Does it have any implications for shift prediction?
Yes, but probably small. I don't know that I've ever seen a paper on shift prediction where people even bring the topic up of the dynamic frequency shifts. I don't know. So if we lump the dynamic frequency shift into H0, we can then write a nice compact expression. We have the usual Liouville-von Neumann equation here that gives the positions of the lines in the spectrum. And then gamma hat is called the relaxation superoperator. That drives relaxation, and the relaxation superoperator is shown here. There's this power spectral density function. There's the double commutator. It could be this factor of a half depends on, again, whether you think the interval goes from 0 to infinity or -infinity to infinity. So again, you have to pay attention when you read a paper. What do the authors mean? The autocorrelation function is even, so we can extend the integral to -infinity. So you just have to be careful. I mean in these notes that the interval goes from -infinity to +infinity, so we have to multiply by a half to correct for that. And again, there's a matter where all these -1s to the Q power come in. So this is nice. This hides all the complexity inside a symbol. A good thing for you is these double commutators have all been worked out by generations of graduate students before you. So unless you want to do some just to see how it works, your main interest is going to be what's the power spectral density function given what I know about my molecule or what I would like to learn about my molecule. So this is an operator equation, and it's frequent. There's nothing wrong with it. This is fine. This is the major result of the theory. But if we want, we can turn it into an algebraic equation. We can expand the density operator in a set of basis functions and then solve for the coefficients, the numerical coefficients. and convert the operator equation into an algebraic equation. And that's, of course, much easier to program on a computer if you want a computer to solve the equation for you. So I write the density operator as a linear combination of basis operators with some coefficients. I do the same thing here. I do the same thing here. Again, we pre-multiply and take the trace. So on this side, the summation is going to collapse because of the orthogonality of the basis functions. And then here, we're going to end up with some trace operation that we have to do. So now we have an algebraic equation. Everything that has an I in front of it is going to look like some kind of generalized frequency. They could be chemical shifts, it could be J couplings, but everything with an I means frequency at the end of the day. And everything that's a real number is going to be a relaxation constant of some kind. So if R and S, for example, were Z, we would think that that's R1 relaxation. Auto relaxation, same operators on both sides, that's R1. If these are Z's but different nuclei, I would say this is the rate constant for IZ magnetization cross-relaxing and becoming SZ. And that's what we think of as the NOE, the nuclear overhauser. So everything is here. If the indices are the same, it's auto relaxation. If the indices are different, it's cross relaxation between different coherences. So here then is the definition of the frequencies, the generalized frequencies. We have the commutator with the initial operator and then the final operator. Here's the definition of the rate constant, the superoperator acting on the starting operator, and then the trace with the final operator. The divisions here are just to normalize in case we don't have the operators normalized to unit. Because now we want the rates to be right. We don't want the rates to be 100 times larger than they are because we didn't normalize properly. And then here's the full expression when I plug in all the doubles. And again, we know the spin operators, we know the basis operators. Everything in these brackets has already been calculated. Our focus again is on the J's. Any questions online? Now, there's a bit of a problem. So let's do a sample calculation. So suppose we have some part of our fluctuating Hamiltonian that depends on A2P0. Remember, P has to keep track of the difference between operators with the same frequency. So for example, I say A2 minus 1 is an operator I minus S plus, A2 0 maybe is 2IZSZ, and A2 plus 1 I plus S minus. So these all have the same coherence order, but the eigenfrequency here is omega I minus omega S, 0, omega S minus omega I. So these have different Z rotation properties. If I'm talking about, say, nitrogen, or not even do that, let's just talk about amide hydrogen and alpha hydrogen. Those frequencies differ by a couple of, let's say, 4 parts per million. On a 500, that's 2 kilohertz. Now, here we can calculate the R2 of the I spin. R2 of the I spin is how does I plus magnetization decay? I have I plus here, I have I plus here, and let's find out what happens, how much relaxation is caused by the term in the Hamiltonian that depends on 2IZSZ. So I'm going to plug in for the A's 2IZSZ, and for BR and BS, I plus. That'll be how much relaxation is caused by that part of the Hamiltonian. So I plug in, I do the commutators, I do the trace, and I find that this part comes to be +1. So that part in brackets is all going to be +1, and I'm going to have some contribution from J0, because the frequency for 2IZSZ is 0. So there'd be a contribution from J0, fine. What about if I put in for one of these operators 2IZSZ, and for the other operator, one of the other Q equals 0 term. So I've done that here. You put I minus S plus for one of the operators A, and 2IZSZ for the other operator A, and I chose the starting operator to be I plus and the ending operator to be S plus. Again, I know what I want to show you, so I knew to pick these. But again, I do the calculation and I get -1. So that also suggests there's going to be some kind of relaxation effect. This isn't 0. And I would interpret this then as cross relaxation between S plus and I plus. Just like I would interpret cross relaxation between IZ and SZ as the nuclear Overhauser effect. Here, I would interpret the result of this calculation as being that there's cross relaxation between I plus and S plus. Now, probably few of you have run a cozy, a homonuclear cozy, but you've probably seen pictures of them in papers. All the peaks you've ever seen mentioned in a cozy spectrum come from the J couplings, not from any kind of cross relaxation. So there's something wrong. Our theory says, a cozy, we generate I plus and S plus and let the J couplings allow. And that's where the cross peaks come from. But our theory says, in addition to the J coupling cross peaks, there should be cross peaks that look like NOEs. And I can assure you, if you pore over the now almost 50 years of cozy spectra, you won't find them. Well, with some exceptions. You generally won't find them. They're not there. So something seems counterintuitive to our experience. If we went through our operator basis, if we have 2 spins, and the operator basis has 16 operators in it, I plus S plus, I minus S minus, so on and so forth. So there's potentially 16 by 16 auto and cross relaxation terms. If every operator can cross relax with every operator, we would have a maximum of 256 possible relaxation pathways. But our experience is that most of them are not there. And the question is, why aren't they there? Our theory that we've just worked out, and I assure you the theory is right, says that they should be there, but they're not. Everybody understand the problem? Okay, so suppose this was right. If this was right, we would say we would have the chemical shift of the I spin, the chemical shift of the S spin, the R2 of the I spin, the R2 of the S spin, and then this cross relaxation rate that came from this calculation in which we had the different terms there. So we can solve this differential equation and see what it predicts. So here's the solution. It's a 2 by 2 matrix. I can write down an analytic result. And here, let's forget about, let's just look here. So what you're going to see here is I've assumed the spins are degenerate, I and S at the same frequency. Let's see if this will run. Okay. I will get it. Yes. No matter how many times I trust the document, it doesn't do anything. All right, where am you? And try to get these to run independently of each other. Okay, so let's just watch the first one here. The chemical shifts are degenerate. Delta omega is the difference between the chemical shift of the I spin and the S spin. And I can just run this one. You can see what happens. I'm on resonance, so you just see decay. I assumed that the initial transverse magnetization of this pink spin was 0, and I excited the blue spin. Of course, if they're degenerate, you might ask how I did that, but this is a simulation, not reality. And then you see the decay. So you can see magnetization builds up on the pink spin. Magnetization is cross-relaxing from the blue spin to the pink spin, just like our theory suggested. The black line is the independent R2 of the blue spin. And the blue line is the bi-exponential decay of the blue spin because it's exchanging magnetization with the pink spin. So this looks just like a nosy buildup. Now, here I move the resonances a little apart. Delta omega is not 0 anymore. So now the pink spin rotates a little bit. It's off resonance. The blue spin rotates. I must have the carrier set in the middle between. But the important thing over here is you can see now the behavior of the pink spin is a little oscillatory. And the decay of the blue spin is still a little perturbed, but it's a lot closer to the black line. And now let's move the spins a little further apart. The oscillatory dependence of pink spin is now more obvious. And the blue spin is almost unperturbed. Its decay looks almost mono-exponential with its pure R2. And then move them further apart. And now the oscillations are very fast with the pink spin. And now the blue spin is almost completely single exponential. And you can see with the pink spin, there's no net buildup of coherence. Some magnetization sloshes or coherence sloshes over from blue to pink, but then it sloshes back again. So that there's zero net buildup. So what does this mean? Where did the markers all go? I don't have a blue marker. This will have to do. So,
You want some from the other side?
no, this is fine. You can imagine this is blue. When the frequency difference is very small, and if these are precessing, they're pointing in the same direction. And cross relaxation is happening from blue to red. And that's what we saw in the first two examples, basically. But as the frequency difference gets larger, the red pen starts to precess relative to the blue one. They both might be going around together, that's irrelevant. The important thing is there's a difference in frequency. So over time, a phase difference builds up between the two magnetization components. Oh, I'm sorry, online this is probably hard to see.
Go back.
Yes, it's hard to see. I was wondering if camera can be focused.
Maybe I need to show myself so I can see. No, maybe not. Let me just move it. Oh. Okay, so you have to imagine I'm holding them horizontally. So when both magnetization components or both coherences are pointing in the same direction, cross relaxation is happening. So coherence is being transferred from the, in this case, black pen to the red pen. But when the frequencies aren't identical, we start to get a phase difference between the red pen and the black pen due to the relative shift difference. And when they become anti-parallel, the cross relaxation starts flowing in the other direction. So coherence is going from black to red, black to red, black to red, black to red. When they get anti-parallel, it's red to black, red to black, red to black, red to black, red to black. Now they're parallel again. Black to red, black to red, black to red, so on and so forth. So when the precession, and that's when you see the oscillation. The oscillations happen when the two coherences become anti-parallel to each other. and the direction of of coherence transfer then changes. And as the difference gets larger, this inversion happens faster and faster. When the shift difference is larger, the phase difference accumulates faster. And in the limit, where the lines are very different in frequency, you have the last example where the reversals happen so quickly that you can't even tell they're happening. And the blue spin just looks like a single exponential decay, and the pink spin just looks like it's sitting at zero, that nothing has happened. So where did this come from? Now, if we go back a second, so here's just the picture again, that when the shift difference is near zero, we see magnetization exchange. And as the shift difference increases, the relative phase evolution gradually quenches the exchange of magnetization because of this in phase and out of phase property. Where did this come from? So if we look back at our equation, the superoperator predicted cross relaxation, but it was quenched by the normal evolution of the chemical shifts due to H naught. So the superoperator predicted cross relaxation, but the effects of the cross relaxation were quenched by the relative phase evolution. And that relative phase evolution came from this part of the equation, the commutator H naught with sigma. That's why we didn't see it in our analysis. Our analysis was fine, but then there was the second part, that once we're back in the lab frame, the relative shift precession has a huge impact on the results. And as a result, all the cross-relaxation pathways that our equation predicts are quenched unless the relevant coherences are essentially degenerate in the lab frame. If they're not degenerate in the lab frame, the relative shift precession is going to kill the cross-relaxation that we predicted would be there. Is that clear? Joshua is looking a little quizzical.
I'll just answer the question that you first asked about why we don't see the relaxation.
Right. Well, the amide hydrogens and the alpha hydrogens are separated in frequency by 4 kilohertz. So as a result, they're zooming around relative to each other, and that cross-relaxation pathway is killed.
So you don't see cross-fertilization, but does it sense with able to tell you that that's the
Or
but it's
where you have two operators that have degenerate or very close to degenerate eigenfrequencies.
sense.
So IZ and SZ, sure, but in other cases it has to be spectral. Most of the other, these 256 terms that are possible, most of them are just killed. For example, single and double quantum coherences are never going to have the same eigenfrequencies. Now, there's also symmetry things that come in. Some of the terms are blocked because of symmetry of the Hamiltonian. And sometimes group theory is brought to bear, particularly in complicated spin systems like methyl groups. So you almost never expect to see cross-relaxation between different coherence orders. 0 quantum and double quantum, or 1 quantum and 2 quantum. But within, say, the -1, Q equals -1 manifold, it'll depend on the particular operators. Do they really have the same eigenfrequencies or not? Then you could wonder how different these things have to be. It turns out by the time the lines are resolved, that's enough. So if you see in your spectra resolved lines, they're not cross-relaxing with each other. Now, Frank Annett, who was at UCLA, I think, for most of his career, in fact, made a habit of measuring these effects. So in a small molecule, if you have resonances that are very, very close to each other, but in a small molecule, the lines can be sharp, so you can still resolve them. You can see these peaks. They're real. The theory is right. It's just that generally you don't see them unless you work really, really hard, and you're not going to see them in a protein. The lines are too broad. But they're there. They're just killed by this. But this leads to something else that I think really important. I think I quizzed you. Well, I don't remember if I quizzed you, and you might not remember the answer if I did quiz you. But when we did the first problem, I think Anne prompted us to recognize that the adiabatic part of R2 was just chemical shift precession. And therefore, we would think a spin echo would get rid of chemical shift precession and therefore get rid of R2. And then we worked out why that's not true. And why was it that that was not true?
Stochastic. It reverse the.
We can't reverse it fast enough here, at least for tumbling. Right? Relaxation dispersion isn't, maybe we can. But there we realized that an implication of the theory was that fast enough spin echoes could perturb the apparent relaxation. Now here we have kind of the opposite effect. Chemical shift evolution quenches the cross relaxation, but you know how to refocus chemical shift. How do you refocus chemical shift? With a spin echo. And again, this is after the stochastic process. We're back in the lab frame. This isn't the nanosecond tumbling anymore. This is on our time scale. So now we potentially could apply 180-degree pulses fast enough that the spins never become anti-parallel. As long as they're, as long as the spins are doing this, so that before they get 180 degrees apart, we refocus them, we would expect to see cross relaxation reappear. And we would see peaks in the cozy that looked like an OE peak. Anyone know what that experiment is called? If you want to see cross peaks from dipole coupling between transverse single quantum operators. Again, you're also young. You might not have heard of it. It's called a Rosie experiment. We don't do it typically just for power handling reasons by doing spin echoes. We do it by turning on a continuous wave RF field. And now the spins are spin locked, pointing in the same direction. And since they're spin locked in the same direction, they're going to cross delay. Of course, in the spin locking frame, there's a difference in tilt angle between the two. So you don't necessarily get the full cross relaxation. It's scaled down by these trigonometric factors. But the main point is we make them cross relax by suppressing the phase evolution caused by the chemical shift. And then these terms that had been forced to zero reappear. When we get to the Trozy experiment, we'll find that there's a cross relaxation between the Trozy line and the anti-Trozy line, predicted by our theory. That depends on the magnitude of the coupling, the dipole coupling to remote protons, the other hydrogens in the molecule, not the amine. So we frequently try to minimize that by deuterating the protein. But even so, the effect would be there. And that would mean that the anti-Trozy line, which is very broad, is transferring magnetization to the narrow Trozy line, and that would degrade the Trozy effect. We wouldn't get the full Trozy effect. We'd be mixing the broad line and the narrow line. That doesn't happen. So in light of what we've talked about, why doesn't that happen? We have, in the N15 spectrum, say, of a protein with the amide, we have the doublet. The doublet is split by 90 hertz. The upfield component of the doublet is narrow, the downfield component of the doublet is broad, and they don't mix. So why don't they mix?
Okay.
The doublet components have the same chemical shift. What's different about them?
Sorry. The J couple.
The J coupling resolves. So there, it's not the chemical shift precession that quenches the cross relaxation. It's precession due to the J coupling. So it doesn't matter where the phase difference comes from. There just has to be a fast enough phase difference. But what happens to the J coupling in a spin echo? It depends on the spin echo, but it's possible to refocus the J coupling. So if we're trying to do Trozy experiments, we have to be very careful about where 180 degree pulses go. Because if we start to refocus the J coupling, then these two lines are going to start to mix and will degrade the performance of the Trozy experiment. Now, in the literature, this whole business is related to what's called the secular hypothesis. And the secular hypothesis means lots of things in physics, and some of that terminology has been brought over into NMR, and it's usually presented as some kind of mysterious thing that we do in the interaction frame. And we'll go back and look at that. But you can remember all the mumbo jumbo about the secular hypothesis and relaxation theory just means that in the lab frame, relative phase evolution will quench any cross relaxation. And I think that's a useful way to think about this, because it alerts you to the fact that the Rosie experiment exists. If I prevent chemical shift evolution, I'm going to see cross relaxation. It warns you about the Rosie effect, about being careful about where you put in spin echoes and how you put spin echoes in. What about the NOE? We said the NOE is always there. Is that true? It takes time to build up.
This Z, I look at the Z, it all resets.
Right? So there's no relative phase evolution, but the spins aren't very smart. It's enough to make them parallel, anti-parallel. So if I want to change two Z magnetizations so that they're anti-parallel, what should I do? How do I turn this one upside down? A selective 180-degree pulse. So if I can selectively, periodically flip this one up and down, it looks like it's rotating in the disco where the strobe lights are going. So you only see it here, 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 but the spin doesn't really, the other spin doesn't know the difference. So you can quench the nosy between two peaks, two spins, by selectively inverting one. And again, we've made them look like they have phase evolution when they don't normally. So this can work both ways. This isn't used so much because it's hard to do selective pulses on a crowded spectrum. But there is an example in nucleic acids called the quiet nosy, invented by Jeffrey Bodenhausen. And there's some group of 3 spins in nucleic acid that Ethan or Joshua can go ask Hashim about and tell us next time, because I can never remember. And you want to measure a weak and a weak between 2 spins that are far apart, but the third spin is kind of in the middle. And there's a huge contamination in the NOE peak intensities from the 2 side hop, going from spin 1 to 2 to 3, rather than going directly from 1 to 3. But spin 2 happens to be in a resolved part of the NMR spectrum. So you can invert it and then see more or less the pure weak NOE from spin 1 and spin 3. But I don't remember which spins these are. You can ask Hashim. But again, it's to drive the point home that this is all really controlled by relative phase evolution. And it doesn't really matter where the phase evolution comes from, or if we kill it, how we kill it. We've done this, we've talked about. So now, if we go back, look at this equation for a moment. Remember here, P and P prime kept track of terms with the same coherence order, but different eigenfrequencies. And we just said if 2 operators have different eigenfrequencies, they're not going to cross-relax. So that argument says we might as well set P equal to P prime and collapse one of these sums. Because anything where P and P prime are different will have different eigenfrequencies. That's what P is telling us. And they're not going to cross-relax. So why bother calculating? Let's just compress the summation right from the beginning. Of course, that doesn't tell us that we could have a rosy experiment. When we collapse them, we've said there are no rosy experiments. We've thrown the rosy experiment out. So one reason I like to talk about what's really happening in the lab frame is to alert you to this idea that maybe we can collapse the phase evolution, or maybe we can create phase evolution. But if we just want to minimize our calculation burden, the argument that the eigenfrequencies have to be the same says that we can collapse and make P prime equal to P. And then when I do that, we end up here. Now we just have 2 sums. We started out with 4. We said Q had to be equal to Q prime because of the rotation properties of the spherical harmonics. And then now we've just said the eigenfrequencies have to be degenerate. Otherwise, cross-relaxation is quenched. So having degenerate frequencies is cause being secular. And when they're not degenerate, that's non-secular, this language that came over from physics. So people would say two coherences cross, so that's only if they're secular, but you can know that means they have the same eigenfrequencies. So most texts that you'll read, the P equal to P prime is done before the transformation out of the interaction frame. And in the very first time, so if you're online or in the class, you're thinking all of this material is so complicated, how am I ever going to learn it? The first time I taught relaxation theory 30 years ago in front of James Keeler, he pointed out that in the interaction frame, you have these phase factors, the E to the I omega T. And there you argue you'll have two phase factors, one from each interaction frame transformation of the Hamiltonian, and you argue they have to, the argument of the exponents have to add up to 0. And that means PS equal to PP prime, and then those phase factors vanish. And the first time I taught in front of James Keeler, he said, when you transform out of the interaction frame, those phase factors are going to cancel. So why do you cancel them in the interaction frame? They're going to cancel anyhow when you transform out. And it took me close to 10 years to come to the understanding I just explained to you. I assume James knew, but he didn't want to tell me. He wanted me to figure it out for myself. But it took about a decade before I realized that it was the coherent part of the Hamiltonian, the commutator with H naught, that was important in the lab frame in quenching the cross relaxation. And what it quenched in the lab frame was completely what you quench by requiring that the phase factors cancel in the interaction frame. So you, as students, can perhaps be happy that this material can take a very long time to understand at a relatively deep level. The other hand, my current understanding of how this works is much deeper spectroscopically than the mere mathematical manipulation of saying just cancel these phase factors. So I learned a lot in the 10-year struggle, and you'll be learning a lot in the next 10 years as well. So here is what would happen back in the interaction frame. We'll have two sets of phase factors because we transform two Hamiltonians, and then we say these have to be 0. Otherwise, in the differential equation, we have some very fast oscillations in the differential equation, and those fast oscillations will average to 0. So the kind of mathematics approach would be to look for fast oscillations in terms in the differential equation, because those fast oscillation terms are going to average to 0 when you integrate the differential equation. But that's a mathematical approach. That's not the NMR spectroscopist approach. The NMR spectroscopist approach is to do what we did, just go back in the lab frame and ask what's happening experimental. So everything in NMR, all the problems in NMR have been solved many, many different ways. Every time somebody stumbles across some new mathematical approach, all the old problems get to be resolved with that new approach. But that can be lots of fun. But the best approach is to be thinking like an NMR spectroscopist. What's the way of solving the problem that gives me the deepest qualitative insight into how the NMR experiment works? And that, I think, is not doing the secular approximation in the interaction frame, it's being in the lab frame. But you'll see this in most textbooks. You'll see in the interaction frame, this expression with two complex exponentials, and then we say, we know that these terms will drive to zero when the differential equation is integrated. So we must require that P and P prime are the same so that this term becomes identity. So here's this leads to what's called the Redfield kite. So for two spins, we have the populations, the Z operators, we have 0 quantum, 1 quantum, double quantum. And in principle, we have a relaxation rate everywhere here. The restriction that Q and Q prime have to be the same, says we won't have cross relaxation between 1 quantum and 2 quantum, for example, so those are all null. And then the requirement that the eigenfrequencies be identical nulls almost everything else. So we have the NOEs up here, and then we have a bunch of operators that just auto-relax. They don't cross-relax with anything, and this is sometimes called the Redfield kite. Here's the head of the kite, and there it's straight. It's tail. Things can get a little more complicated if we have two kinds of Hamiltonian. So if we have, for example, for the nitrogen 15, we have both the dipole Hamiltonian and the CSA Hamiltonian. And when we form the double commutator, so the total Hamiltonian is the sum of both, and we calculate the double commutator, there can be lots of cross terms between the two Hamiltonians. And some of those will survive and give cross relaxation in that case. That's what's indicated by these dashed lines, that there can be more happening when we have more than one Hamiltonian, more than one interaction. And the famous one for the nitrogen is CSA and dipole. And this cross relaxation term that would arise in that case adds to R2 for one of the doublet lines and subtracts from R2 for the other doublet line. So one line gets increased and the other line gets narrowed, which is the Trojan effect. Or in a methyl group, you have three different carbon-hydrogen dipole-dipole interactions. And that will expand what cross-relaxation pathways can occur. And again, that can be manipulated to give the famous methyl group Trojan effect that Lewis K developed. So slightly more complicated, but only really because the commutators end up with many more terms, and the spectral density function will become a cross-spectral density function, because you'll have the stochastic term, say, for the dipole interaction, and the stochastic term for the chemical shift interaction together in an ensemble average to a cross-correlation function. So these are called event relaxation interference effects or cross-correlated relaxation. But they're simple extensions of the theory. It's just that there's more terms to calculate. Let's see. Almost done. The double commutator that I said, the double commutators have been calculated for everything under the sun. But it is possible to rewrite the double commutator by making use of the fact that the trace is cyclic. You can permute operators in the trace and it remains the same. So playing around with that, you can get the double commutator to be a product of individual commutators. And sometimes that's useful, but they're just for you to know that these things can be manipulated. It's fairly rare to see this form in a book. I only know of it because a former student of Ann's, Tatiana Yumanova, who's now a professor at Texas A&M, always put things in this form. And I don't know if she learned it from you or whether that came from Russia. So here's what we said. If we apply a spin echo or a spin lock, we can prevent the phase evolution from chemical shift, and that's called the Rosie experiment. I mean, one reason that experiment is useful is in medium-sized molecules, when we talk more about the NOE, you'll see that the NOE might be zero for certain tumbling times, whereas the Rosie effect is always positive. So in medium-sized molecules, sometimes people run the Rosie just to make sure they're not missing NOE crossbeats. It also turns out that chemical exchange crossbeats in the nosy have the same sign as NOE peaks in the real NOE peaks in the nosy spectrum. So it can be hard to tell them apart. But in a Rosie, the chemical exchange peaks have opposite sign from the ROE peaks. So sometimes people run a Rosie in order to check, do I have any chemical exchange cross peaks? And then because for macromolecules, they have opposite signs, it's possible to average them together and cancel the cross relaxation effect. So there are experiments, for example, in the epoxy experiment, you can get artifacts from cross relaxation as well as J coupling evolution. And by balancing the nosy and Rosie effect, you can suppress those so-called clean epoxy experiments. Quiet nosy, next time someone from Hashim's group will tell me what I add here to indicate what spins this experiment can be used for. And then, of course, Rosie is incredibly important these days. And if you go and look at, for example, a Rosie-based relaxation dispersion experiment, for example, the Rosie CPMG experiment that Pat Lauria developed, a CPMG experiment is a block of spin echoes. So this is really bad. And you'll see in his sequence, in the middle, one of the doublet lines is inverted relative to the other in order to give them a phase difference. Now, you might think we need to do that multiple times. When Pat first thought the experiment up, we did it multiple times so that the spins were toggling. But there's too big a relaxation loss doing that. And eventually we realized in most cases once is enough. It's a fairly slow effect. You just have to toggle faster than the cross relaxation builds up very far. I mean, you'll see it's there just for that effect. This is when I said if we have more than one interaction, we just have to sum over all the interactions. And we end up with the auto relaxation effects. They just the dipole contribution, this plus the CSA contribution, and then the interference, the cross terms. And in the cross terms, we have a cross spectral density function. And here we have just the normal power spectral density function. Yeah, I think next time we'll start with the examples. So we'll take a break and then do problems, I hope. Any questions before I turn things over to Anish? We'll take a 5-minute break, or at least me to get coffee. Any questions online? Okay, well, you can review the notes, they're online, you can ask more questions tomorrow.
Go first.
Alright.
Let me just share my screen and make sure. So today we are going to be talking about problem set that we were kind of supposed to do a lot earlier because we got busy with solid state NMR. Today is going to be. Oh, I forgot which. This is the one on cozy spectra and HLCO. I think this problem is 3 in our sequence or in something. Anybody attempted it? By any chance? One problem, at least. First one.
Is this the,
Cozy, beta
was
cozy.
the,
Yeah.
I would attempt it.
Thanks for that. I know how to do it.
I have the right Christian who invented the problem,
Yeah.
don't get a mistake.
You did?
Yes. I made me very happy. It's hard to catch Christian in mistake.
I think I should be able to. Yeah. Okay, it looks better. Only now that my computer decides to go for it. Great. So, this problem of problem set 3 So something like this. Or it's a development or sort of a heuristic development of commonly used double quantum filtered cosine for combinatorial correlation, etc. And this starts with the sequence cosine beta. It goes something like beta pi over 2 is 91. Then a pulse of flip angle beta and phase y followed by acquisition. And the first question asks you product operators present prior to T2, the acquisition delay or the acquisition, assuming beta equals 0, pi over 2, pi, 3 pi over. So unlike a phase cycle, you shift the phase of the pulse. Here, the second pulse is of constant phase y in all the steps, but it's the flip angle, so the duration of the pulse is being changed. With a phase, it's a zero pulse, meaning no RF. In the first step, it's a 90-degree pulse. In the second step, it's a 180-degree pulse, and then 270-degree pulse in the last step. Assume that at the start of the sequence, you have two spins, I and S, and I'm just going to use the I and S for simplicity, but assume that I and S are homonuclear. Okay, so they're the same nuclei, they're both protons, for example. And assume that you have telegraph shift omega I and the J coupling. And you start this pulse sequence from I. Okay, I mean, in principle, you will get both I and S. The equilibrium density operator is both IZ and SC. But assume for a moment that you want to start with IZ, because for SC the calculation will be exactly equivalent as long as because they both have one J coupling relative to each other. So I'm going to assume that my time 0 density operator is proportional to IZ. Then I apply a 180 or 90x. What does that give me? Minus IY. But that's actually different. Okay, so in the delay T1, what are the two evolution forms that we see? Scalar double. So let's take it one step at a time. What happens when you evolve telegraph shift? Minus. Do you agree? Is it plus IX or minus IX? X evolves into plus y, y into minus x to minus y to plus x. Plus x sin omega I t1. Then we said you have the J coupling. So what are the four terms that will be produced at the end of T1?
Sine y plus the I is your sine of I J your
Minus or plus?
Plus
Sine I.
So it will always carry the cos, right? Cos chemical shift, sin scalar doublet. Then plus ix
Sign.
cosine
Sign of that is sign.
sine of that chemical shift cosine J coupling and
C.
sine chemical shift sine J coupling. It's okay. So this is the part that is the same for all four sets. Experiment. I'm going to sort of develop a heuristic, right? So, going to take this experiment forward. So I'm going to write this as minus IY 2IXSG IX 2IYSV. And the parentheses just denote the trigonometric terms that will carry in every step. So when beta equals 0, I just have this at the start of acquisition. Right? That makes sense? Because there is no rotation of any of these stuff. What happens when beta equals pi over 2? And keep in mind that it's a y phase pulse. It's a y phase 90-degree pulse. And what happens to each of these four terms when you do that?
I wanted to say
Next one. IX
S.
Are you sure? With a y rotation, v goes to plus x goes to minus y. Sorry, plus x goes to minus v. So that obviously gives you minus iv over here. And what happens to 2IYSV? Yes. What term? Well, is it only double quantum?
Oh, zero.
The mix of double and zero quantum coherences. And so that what form does it take here? Okay, that was the first step. Next one is perhaps faster. So 180y, this again is going to be invariant in all steps. This experiment. What happens to this term? And always remember this is our baseline. This was first step when you're applying the rotations on the terms that came on the first step. Okay, so 180y on 2yxxv. Right. Ix 2yxv. Minus 270y is equivalent, of course, to 90 minus y. So what does that produce?
Minus 2IZSS.
Here you still get a minus. XV. Here you get an IZ. right? And the last one is negative. Is this okay? We got past, I think, what is part 1 or part A, yes. For product operators prior to acquisition. And the question says find a receiver phase cycle. I found this wording a little bit unusual because I'm not exactly varying the well, you could call it a phase cycle, even though you're changing the pulse duration every single time. You're doing a 0 pulse and 90 and 180.
No, the receiver phase cycle is the adding is.
Yeah. So add and subtract the experiments in such a way as to isolate the product operator representing a diagonal coherence and the product operator representing a cross peak of diagonal coherence. It has the same line shape. So we're looking for absorptive doublets on both the diagonal absorptive signals, both the diagonal and the off diagonal peaks. Because you don't want the trouble arising from traditional cozy, where the diagonal is faithfully absorptive, the off diagonal will be dispersive and vice versa. So, any ideas how you can add and subtract combinations of these 4 experiments in a manner that you produce only, in a manner that you get terms which are both absorptive, diagonal peak and off diagonal peak. Out of these 4, one is obviously irrelevant at the start of acquisition because it contributes no detectable signal. One is that. This one, no detectable signal.
Inside of, if you scroll up a little bit, where we can see the trig functions. For example, IY, what was its frequency during T1? What's this chemical shift during T1? Pardon?
Is the answer?
Right, it's cosine omega I. And at the end of the experiment, this IY, what's the frequency going to be? Okay, so that's a diagonal peak. So if you scroll down, maybe where you have your parentheses, where your beta equals 0 line,
Yeah.
why don't we put a D there in the parentheses for the IY? So we know that's a diagonal. Let's look at the 2IXSZ, go up to the trig terms. Do IXSZ, what's its frequency during T2? Pardon?
Omega I.
It's omega I because it's IX. And what's its frequency during T1? But it's also a diagonal peak. We'll put a D there. And back up. The IX term
It's also diagonal.
oh no, I'm sorry, we have to look, I'm doing the wrong thing.
So, and the IZSS will involve T2 already S. Is that what you're saying?
This term,
Yeah.
Yeah, let's get that wrong.
that term. These two are the off-diagonal coherences.
Those are the cross.
Correct.
And then the diagonals. I mean, the terms at the end don't matter. The IZs are the capital. If we look at the IX term, what was it during T1 if you scroll back up here? The IX term is a diagonal term. And then the IY term is also a diagonal.
And you know, if you can range for, you can allow it.
So
One of them or the other.
now we're trying to find the diagonal peak and the cross peak will have the same line shape. So for example, as IY are going to have the same line shape as 2 IXSC.
Or
They're
two,
not, one is in phase and the other is anti.
or even for example, IY and 2IZSX. IY is in phase diagonal coherence and IZSX is but in phase absorptive diagonal coherence, and 2IZSX is antiphase dispersive cross peak coherence. That's what we don't want. We want both the diagonal and the cross peak to be absorptive. So the simplest thing to do is to, yes, you're going to say something? Is to preserve these four and try and arrange for everything else to get zeroed out. You can see that on this column, the IX and you have IX over here and minus IX over here. So if you just added 0 and pi, you get rid of IX. Similarly, if you add pi over 2 and 3 pi over 2, you get rid of IZ. That makes sense? So 0 plus x plus pi, that gives to 0 plus pi. That gives me 2 times minus IY, 2 times 2IXSZ. This term goes away, and that term, the last term, also goes away. Similarly, if I do pi over 2, that's 3 pi over 2. I again get 2 times minus IY, and 2 times then IZ minus IZ goes away, and this will go. Does it make sense? Choice of additions. Now, do we further add? What will happen if we add all of these two also? We just ended up adding everything.
Phase dispersive.
Uh-huh. Yeah, so the IY is going to give you in phase if your phase to give X as absorptive. So if this cross field coherence, you phase it to be absorptive, this diagonal coherence will be dispersive if you added them. So instead, what you simply do is take the difference. Then this also goes away. So you are only left with 4IZ plus 4IZSL. Importantly, of course, in the when you evolve it in the direct dimension, the peaks will be antiphase absorptive. That's antiphase will be creating antiphase coherence. But that's okay.
You don't understand the pi over 2 plus 3 pi over 2. Oh, I see what you mean, the beta.
Yeah.
The beta, okay.
Yeah.
You're adding experiments 1 and 3,
Yeah.
adding experiments 2 and 4,
And
and
subtract.
then subtracting those two.
Yeah. 1 plus 3 and 2 plus 4.
Does everyone see that?
So that becomes your receiver phase cycle.
So what does that mean, the receiver phase cycle looks like? Yeah. A 0,
I guess in Bruker language it would be 0, 2, 0, 2.
a 0, 180, 0, 180.
X minus X.
Experiment 1 and 3 are both positive, and 2 and 4 are subtracted. +1, -1, +1, -1. You broke out the E, that would be 0, 2, 0, 2.
That is the receiver phase cycle. We want to do this experiment by varying the pulse length data.
Let's pause for question.
Yes.
On your final question. Yes. My question first was like just to add a little better. This you remind me, is that second term in your final line, is that the diagonal?
This one?
Yeah,
So which one is diagonal, which one is
yeah.
off tangential? For that you think about what nucleus that coherence belongs to. So we started with I, and so you evolve during T1, the indirect dimension at omega I. If you evolve during T2 at also omega I, then you are a diagonal coherent. But if you evolve during T2 at omega S, you are a cross-peak coherent. So then this one, the first one, 4IXSZ, evolves during T2 at what frequency? Omega I. The transverse is on I. Then 4IZSX, which maybe I guess we are used to writing it other way around. So if you want, you can write it as 4SXIZ. Will evolve during T2 at what frequency? Omega S. That then becomes your cross-peak coherent.
And clearly they have the same line shape in acquisition, but if you look up the column, they came from the same starting operator.
Yeah.
So they must have the same line shape in T1. After T1, they all came from the 2 IXSZ operator. So they must have the same line shape in T1, and therefore they have the same line shape in T2. But notice we took that one operator and essentially split it in half. Half became the diagonal peak and half became the constant. So the relative sensitivity went down by a factor of 2. So we sacrificed the power of 2 in return to have the same line shape. No free lunch.
Sense. It's
Okay, what was the next one? We have previously shown that Ry of beta, and this is, if you think about, I think I like to use rotations in space, think about rotations in order to make sense of these formulas. Ry of beta is a rotation about y-axis through an angle beta is given by this R Z90, Rx beta, R minus Z90. And we are coming to this after doing solid state NMR. So we should, I guess, have reasonable intuition about moving between axis frames. So this is like taking, you're trying to do a rotation about the y-axis. And what this formula on the right is saying is rotate about minus z by 90. So y moves to x, then rotate about x through beta, and then rotate back around z through 90 to go back to y. That's how I'm describing a beta rotation, or y rotation through an angle beta by this product of 3 rotations, where I want to describe a rotation around x. So this is how you would convert a rotation around y into a rotation around x by sandwiching these z minus z rotations.
The
And
rotation around minus z by power of 2 is the same rotation around z
z
and
minus
minus
pi.
I think in the notes it's written as the
Yeah.
minus angle, not the negative aspect, but they're the same.
And we probably will come back to this when we talk about Motional averaging of tensors in solid state NMR, where you want to do an X rotation and you want to adhere to the Rolle's convention for your Euler angles. So an X rotation can similarly be written as Z by Z rotation, the appropriate angles. But what we are going to do is essentially try and rewrite this pulse, which is the variable flip angle. Right, so you're doing exactly for almost like 4 different pulse programs, and instead rewrite it as a pulse program where the pulse durations are fixed, but only the phase of the pulse vary. And to do that, we need to prove this relation. This is starting again with writing y rotation as this. Instead of the zxz, I'm now writing it as a minus xzx. Is again equivalent because I'm rotating around x to bring y to z, then defining a rotation around z, and then bringing it back down y. And I need to prove that this is equal to this. Any ideas on how to do that? I need to prove that this is equal to that.
Use like a conjugation relationship, and it's switch the order of the two.
These two. Right. How would you do that? on the right track. Simplest thing you should think of is this last term is sort of staying there or not changing. And if you see there is an RZ beta here on the left side, the easiest thing is to multiply by RZ beta and then RZ minus beta. Right? Because this is unity or identity. This is rotate to Z and then rotate back. It's not doing anything. So now with this, I have the left edge of the expression need to prove is here, the right edge is also here. And if you look at this kind of looks like a unitary transform, right? Now, if you think of the xy plane, and you think of an axis going off like that, an angle beta relative to the minus x. Does it make sense that a 90 rotation around this axis can be represented using this expression? Right, because I'm taking this axis, which is that beta relative to minus x, rotating through beta by around z, so I'm bringing it to minus x, then doing a 90 rotation around minus x, and then bringing it back. So this, and this, and back here. Is this making sense? Any questions? Or was this too, I don't know, non-intuitive? So there are still some physical loops. So questions, please.
You just heard this rotation starting.
I'm just considering this block here. Okay. What is this block describing? So it's describing like rotate around z through beta, then rotate through minus x, or pi over 2, then rotate back around z through minus beta. It's kind of like flip through z through an angle beta to arrive at minus x, then do a 90 rotation around minus x, and then go back. So that means that the axis that is being described is at beta relative to minus x. Does that make sense? That's what this is. It's an axis which makes an angle beta with minus x. And when you do a z rotation to beta, you bring it to minus x. Perform the rotation pi over 2 and then bring yourself back. That describes the 90 rotation around this axis. If that was not intuitive, then you can also you can apply like regular angular momentum algebra and prove that this is uh, actually what I'm describing. So this expression becomes this is and I need for the main rotation I need to describe an angle through which I rotated and the phase of the axis. Right? The angle through which I want to do the rotation is 90. That's what I did. I did a 90 rotation around minus x and then brought myself back. So what is the phase that I would describe?
Looks like 180 minus beta. Plus beta. I can appreciate it too.
Right, because theta is 90, then I land at plus y. And therefore it should be 180 minus beta.
I have to say, so. So the z rotation of beta is minus x.
And we have to read this from right to left. This is an operator gradient. So first we rotate counterclockwise from my beta. And that brings the axis back to the minus x. And we rotate around minus x. And then we go back. So it's a pi over 2 pulse oriented at the angle beta relative to the minus x axis. But that means it's 180 minus beta.
Yeah.
Measured from the plus x axis. Is the phase of the pulse. Described by that sandwich.
That is only not what you write here. It might be plus beta. I'm trying to discern if I got this the other way around.
Can I see the equation again?
This one?
Yeah. Uh,
So this phase actually may not end up mattering. If I try to do this algebraically, then the term of interest is does everyone follow this? Follow what's going on here? I'm trying to arrive at the same expression for the phase of the resulting pulse using just operator algebra. So R minus X and rotation can be represented as exponential. And this is the unitary sandwich, U inverse U, an operator sandwich in U inverse U. So I can then take that inside the exponent. And then I should get just give me the this is what you don't remember. Just give me minus y or plus. I think this should give minus y. Then when you account for an extra minus outside, you get minus x and plus y, which does land you here on the xy plane. So maybe we have to check this. Yeah, maybe we need to double check. I think ultimately for the experiment, this wouldn't make a difference at the end of it. But we still need to confirm that. Anyhow, for now we'll just say that. And let's assume the answer given the problem, which is 180 degree plus beta. Does this make sense? Both the way of intuitively deriving the axis and algebraically arriving at the axis. Both of those approaches are important. Okay, I'll just show you how to convert this to a pulse program, and then you can construct the phase cycle by yourself at home. This is the final operator, right? We replaced that one pulse, which was the rotation around y through an angle beta. That's where we started. Ry beta is written as this sandwich, which is written as this, and that's what we proved. So I replace this one beta y rotation with this sandwich. So how does the pulse program now look? Starts of course with a pi by 2 x, and then a t1 for the indirect dimension, and then you have this beta y. Now that gets replaced by this. So what does that look like? And remember, you lead from the right. What is the first rotation? 90x pulse. And then the 90 pulse with this phase 180 plus beta. Right? There is no time gap between these two, just spacing them out. So we got that. Then what? Rx pi over 2, R90 plus beta pi over 2, and then what element is that? What does RZ of beta? Receiver phase. So you turn on the receiver and you set the receiver to phase minus beta. That will then account for the Z rotation around beta. Now you know what the four values of beta are, right? 0, pi over 2, 3 pi over 2, and 0, pi over 2, pi, 3 pi over 2. For those four values of beta, you can calculate what the phases of this pulse should be. And then in each step, you can calculate what the receiver phase should be. And that then becomes a 4-step phase cycle for a double quantum filter cozy. Make sense so far? And you could also prove to yourself that this is a double quantum filter. Because for a 4-step phase cycle where you increment the phase by minus 90, because that's what's happening to minus beta. Right? Beta goes up in steps of 90. So minus beta is also rotating to minus 90. The receiver phase is also going to do that. So what values of delta P will you select for in this phase cycle? And therefore, before those pulses, what coherence will you pick up? We have just enough time to see if anybody has the answer. Okay, if not, then we will come back to that next time. You can review the notes and check what values of change in coherence order will be picked up in a 4-step phase cycle.
The state of magnetization there.
Sorry,
State of magnetization during that block.
which block?
I over to is still
No. Which one? Like P or the start of delta P? No. You do pick up plus 2 and minus 2 coherence before that second pulse. Like, you know that at the end of during acquisition, you will pick up minus 1 coherence. That's a selection rule. And then the delta P again is set by your phase cycle. So you can arrive, you will get the answer that you pick up plus 2 and minus 2 coherence before you produce plus and minus 1 coherence, and then they will become plus 2 and minus 2 coherence. Then the start of acquisition, you pick up the minus 1. As soon as this is, you can look at the notes and you can see that this will be consistent. You have to get a delta P of either 3 or minus 1. Anyhow, I think we have to leave. Hope this was helpful, and we can review this again next class.